And then I'm going to broadcast.
Okay. All right, everyone, we are going live. All righty. Okay. All right, so we are live. Welcome, everyone, to the first webinar of the Ocean Acidification Hypoxia Science Task Force. I'm going to share my slides. 1 second. Share screen. All righty. Can everyone see that?
Yep.
Start from the beginning. All right. Okay, here we go. Alrighty, so thank you for joining today. This is the webinar of the Ocean Acidification Hypoxia Science Task Force. The webinar topic today is U.S. West Coast Ocean Acidification Observations and Assets Inventory. Starting at 1 p.m. We will be going till about 3. Not sure if we'll use all that time depending on the discussion, but wanted to leave ample time for people to discuss this topic. So I am Haley Carter. I am a program scientist at Ocean Science Trust based in Oakland, California. And I'll be hosting this webinar on behalf of the task force with funding from the California Ocean Protection Council. And Richard Feely, member of the newly convened task force, is going to be moderating the discussion today as well, and also kicking off with the first presentation. So just a little bit about housekeeping. This webinar is being recorded so we can document it online and also share it with folks who are not able to be here today. And if you have a question throughout any of the presentations, we're going to save questions until the end, but please feel free to chat a question at us. We will get to those list of questions at the end. So you can chat those in at any time to Ocean Science Trust. Participants are logged in as listen-only mode, and we have some of the task force members logged in and they're able to mute and unmute themselves. So when you're presenting, unmute yourself, and then when you are not speaking, make sure to mute yourself so we don't get any feedback. And Dick Feely will be posing 5 questions to the task force members at the end, and then we will open it up to the public for questions. Alrighty, so the purpose of these webinars, this is, as I mentioned, our first webinar in the series, and on behalf of the task force, we are hoping to track scientific efforts related to ocean acidification hypoxia and coordinate across the US West Coast region and beyond. This is also a forum for engagement between scientists and decision makers, so we have a lot of folks on from state and federal agencies, and also a forum for technical discussion about ongoing unpublished research. So largely state-funded efforts, but wanting to track along as these efforts are going forward. So a little bit about our audience. I wanted to share, these are the registrants. So a lot of folks from California, this is the location by percentage, and also lots of science folks, state, federal government, NGOs, some tribal representatives, and some industry folks. So just a little bit about the OAH Science Task Force. This was convened earlier this year to serve as a responsive advisory body to the Ocean Protection Council to provide scientific guidance to inform continued action on ocean acidification hypoxia in California and along the West Coast. This was convened in response to recommendations of the previously convened West Coast OAH Hypoxia Science Panel, who came out with recommendations in 2016, and also in response to a California Assembly Bill 2139. This group is providing scientific advising to inform future actions in California, and also is providing scientific guidance into California's ocean acidification hypoxia action plan to be released later this year as part of our membership in the International Alliance for Ocean Acidification. All right. These are the task force members, so they represent membership from the West Coast, interdisciplinary group. Also, some of the members are previous West Coast panel members. It's also co-chaired by Steve Weisberg from the Southern California Coastal Water Research Project and Francis Chan from Oregon State University. Some of these folks will be leading other webinars, so stay tuned for those. We're hoping to have these webinars every 2 months or so. And so today our topics are going to focus on, Dick Feely is going to provide an overview of anthropogenic CO2 along the US West Cost, where they're going to pass it off to Tessa Hill from University of California, Davis, who's going to narrow in a little bit more on California. And then Sarah Braley from the Ocean Protection Council is going to talk about the Joint Ocean Acidification Hypoxia Monitoring Task Force, the status of California's monitoring assets and inventory. And this is actually a West Coast inventory. And then we'll pass it on to Karen Brady from the Oregon Department of Fish and Wildlife, who is going to talk about where we are headed as a region, identifying gaps and priority investments for the West Coast. And then we will have a moderated discussion by Dick Feely with members of the task force, and then we will open it up to public questions from the public. Just a bit about today's speakers. Richard Feely is a NOAA Senior Fellow from the NOAA Pacific Marine Environmental Laboratory in Seattle, Washington. He holds an affiliate full professor faculty position at the University of Washington School of Oceanography. His major research areas are carbon cycling in the oceans and ocean acidification processes. He's also authored more than 300 scientific research publications. He's currently a member of the U.S. Interagency Working Group on Ocean Acidification, and is also a member of the newly convened OAH Science Task Force here in California. And he will be moderating our discussion. He's joining us from Poland, so I appreciate that, and it is nighttime there for him. So we will pass it off to him. I'll introduce our other speakers as we get to them. And without further ado, I will pass it off to Dick. So Dick, just feel free to share your screen, and I will stop sharing mine. And thank you everyone for joining us.
Okay.
So we see the end of the slideshow, so you might have to go back to the beginning.
Oh, how do I go back to the beginning?
So maybe click escape. Wouldn't be a webinar without some technical difficulties, so thanks everyone for your patience.
I'm going to turn it back to you. How do I do that?
So let's see. Should be able to share the PowerPoint or.
Yeah, I just got to get to my desktop. I'm not getting to my desktop.
Okay, let's see. See if I can. We can try to pull up your presentation as well.
Yeah.
Give us a second.
Before I got to my desktop, now I can't.
Yeah, the interface is a little hard to work with.
Just pull it up yourself, then I'll walk it through.
If you go to view options in the top, you should be able to exit full screen mode, which might be something, but we will share our screen as well.
Let's see. Oh, I see.
There we go.
Oh, there it is. Oh, is that mine or yours?
This is ours.
Okay, yeah, just tell us when to advance the slides.
All right, you can start. Pull it
Okay.
up.
Great, there we go.
Okay, welcome everyone. It's really a great thing we're doing this today, and I'm very excited to be here. What I want to do is to give you a satellite's eye view of ocean acidification along our entire coast. And I'm going to focus in on two issues. The first one is what is the distribution of anthropogenic CO2 and what are the changes over time. And the second issue I want to bring up was how the changing buffer capacity of the ocean affects the changes that will take place in the very near future. And this will be true for the entire region. So it'll be a really good indication of what we might expect. Next slide, please. So this is a map of the Pacific Ocean. You can see North America in the top corner, and we're looking at, say, from Antarctica in the southwestern Antarctica. And what you're looking at is a time series of the accumulation of anthropogenic CO2, that is the increase of dissolved inorganic carbon over time from 1994 to 2007. The yellow colors you see is the depth penetration of that anthropogenic carbon along the transit lines that we've occupied. We've repeated this now 3 times in the Pacific Ocean, and that's allowed us to determine how the anthropogenic carbon is accumulation in the ocean. right along the Pacific coastline, you can see our sections. We've done these sections now 5 times. And so we can see that buildup of anthropogenic CO2 along our coast. I'm going to zero in on that coastline in the next few slides. On the right-hand side is the very famous Mauna Loa Aloha Station record of the buildup of PCO2 in surface waters. And you can see that we have now enough data to see the trend of the increase over time. And that's about right around 2 to 3 microatmospheres per year. Next slide, please. So what we have done to develop this observing system on the West Coast is combine all the assets we have within the federal government along in collaboration with our state and local governments in order to have an observing system that's fully integrated. So we do this through the IWG observing system, and we place the CO2 measurement sensors on the moorings, and we collect samples from cruises and surface observations, in some cases wave gliders, and we collect all this data and we combine it all together so we get a picture of the changes that are taking place. And this right-hand panel is an example from Monterey Bay, the Francisco Chavez, beautiful long-term data set of the changes in PCO2 in the surface waters in the top panel and the change in pH in the bottom panel. And what we have overplotted on that is the Mauna Loa record. So you can see very clearly that while we have much higher variability in PCO2 and pH in the coastal waters as opposed to the open ocean waters, when we have a long enough record, we can see the long-term trend. And you can see in the pH, for example, the red colors of the pH is the changes at the hot station as compared to the change in Monterey Bay, which is in blue. And what you can see is that the rate of change is a little bit faster than what we see at the hot station. And that's because the acidification that's occurring, that rate change, is a combination of the atmospheric source of CO2 plus an upwelling source plus a source from the nutrient sources and other rich sources that add to the acidification. So in our coastal region, we have a little bit faster rate of acidification, and with these records, we can actually detect that. And from our work from observations and modelings, we now know that the anthropogenic carbon consists of large-scale global sources plus local sources that we would have from industrial and natural regions and urban regions. And we can see the changes in the chemistry resulting from both. And our models are showing this quite well. Next slide, please. So this slide shows the corrosive waters, and in the right-hand panel, it was the corrosive waters, what we observed on the first cruise in 2007. The red colors are when that corrosive water gets very close to the surface. This means the saturation state for aragonite is less than 1, and we can see in 2007, we found some places where it was right at the surface. And when you look at, on the left-hand panel, the 2016 data, you can see that we see much more corrosive waters to the surface along our coast. This is a combination of the long-term changes that are taking place because that corrosive water migrates on the average about 1 to 2 meters per year towards the surface, plus the local seasonal changes in the rate of uptaking. But we can easily see that we have very corrosive waters very close to the surface and impacting the biology on our ecosystem along our coast. Next slide, please. Switch to the next slide, please. So this slide shows what the anthropogenic CO2 looks like along our coast. And the left side is the surface And then I've also shown on the right-hand panel is the 50-meter depth. And what you see is that there's a little higher concentrations of anthropogenic CO2 in surface waters to the south, about 60 to 65 micromoles per kilogram. And as you go from south to north, that concentration decreases a bit overall. In the northern regions in Oregon and British Columbia, it's about 55 micromoles per kilogram. This has to do with the way anthropogenic CO2 is absorbing from the atmosphere into the oceans. Now, in the central regions, you can see this clearly in that the 50-meter depth, the lowest concentrations are nearest the coast. And this is because you have the upwelling having its impact right near the surface, and the strongest upwelling is along the coast there. And so when you mix that water with a lower anthropogenic CO2 content, about 30 micromoles per kilogram, with the 60 micromole per kilogram surface waters, you end up getting a value around 40 to 55 micromoles per kilogram. The important take-home message on that is that throughout the entire water column, you have a significant amount of anthropogenic CO2, which we can determine what that concentration is, and it is adding to the upwelled water, and so the sum of the upwelled water and the anthropogenic CO2 is what is impacting our organisms. In surface waters, the dominant component is the air-sea exchange of anthropogenic CO2 across the surface. In the subsurface water, it's a combination depending on what depth you're at. Next slide, please. These, what happened?
All right, give us a second. We'll pull it up again.
Yeah, it seems PowerPoint quit on me, sorry about that.
I'm gonna swap the displays again. One sec, sorry about that. There we go. I'm gonna advance a few slides.
There, okay. This is a movie slide of the buildup of anthropogenic CO2 in the water column. On the left-hand side is Southern California and Mexico. On the right-hand side is Washington and Oregon and British Columbia. And again, what I was saying earlier is that you can see that anthropogenic CO2 building into the water column, enriching from the surface down, and you see a little higher concentrations to the south than to the north. But interestingly enough, when you get further north, it actually penetrates deeper into the water column the further north you go. This is due to the deeper ventilation of the water as you move north in along our coast. But it's very clear that you can see throughout the entire region penetration of anthropogenic CO2 far below the shelf break, down to 400 or 500 meters. The shelf break's at 200 meters. So we know from these kinds of outputs that we have anthropogenic CO2 throughout the entire water column on our shelf. The right-hand panel shows the change in the aragonite saturation state that has taken place over time. You can see that changes. In surface waters, it's about 0.5 or 0.6 decrease in saturation state. And again, you can see that penetration down into the water column so that you can see that the change in saturation state from the pre-industrial to the present is migrating down into the water column as we accumulate more and more CO2 from the surface. Next slide, please. This is a slide that shows the rate of change that we are observing. It's about 1.5 micromoles per kilogram to the south. And as you go towards the north, it gets closer to about 1 micromole per kilogram per year. And as you go down in the water column, you can see it drops to a value of about about 0.5 micromole per kilogram per year. So we can see that build up over time and how fast it's changing. It allows us then to get a sense of how fast things are changing. We can use this data to go and include in models and predict the pH changes or aragonite saturation changes with time. Next slide, please. Now this is a indication of how we look at how the chemical changes will take place in the future and what are the drivers. As we add more CO2 to the ocean, we change the buffer capacity, and the buffer capacity is related to us by the Weve factor. And the Weve factor is the bottom panel. And you can see as you add CO2 from 400 to 900 microatmosphere, that Weve factor is increasing and increased much more quickly in the colder waters. So we see more rapid changes in the Weve factor. And the impact of those changes in that decreasing buffer capacity is that the pH changes are much quicker. And so we see more rapid pH changes over time here on the West Coast than we would see in the Gulf of Mexico or other regions where the waters are warmer. But it's interesting that the saturation state changes are slower. So this is the way the buffer capacity affects the changes, and we see opposite effects for pH changes and aragonite saturation changes. And that will have an important impact over the long term. Now, the best way to look at that is in the next slide. The next slide is from a very nice paper by Steve Pacella. And what he shows here is the changes over time in pH from the pre-industrial out to 2100. And what you can see is that the changes are just beginning to start to change. We can see those changes out around 2015. But very quickly, the change in pH will accelerate. We'll begin to have more rapid pH change. And the difference between the minimum and the maximum will increase. That means we'll have higher seasonal variability and a higher diurnal variability because of the changing buffer capacity. This data was taken for Puget Sound as an example. The center panel shows the same effect for PCO2. And what you can see is about 2015 or so, we start to see this rapid change in PCO2 in which the PCO2 begins to go way above 1,000. And 1,000 is the concentration when hypercapnia takes over and you begin to have physiological effects on marine organisms and it affects their behavior. And we will see these kinds of changes taking place. In Puget Sound, for example, we can see that PCO2 will jump up as high as 3,000 or 3,500 microatmospheres by 2100. So these are very rapid changes in the way the chemistry, and that seasonal cycle or diurnal cycle will begin to be very much increased over time. And then in the right-hand panel is the aragonite saturation. And as I said before, it begins to decrease overall in concentration, but the seasonal cycles and diurnal cycles will actually become smaller. And that's because the values are getting so low that their variability is not high. So last slide. Next slide. I just wanted to say that our challenge then is to understand the changes in the anthropogenic CO2 and acidification that causes in association with the biological responses and be able to determine how much of those biological responses are directly attributable to the anthropogenic CO2. So we have to combine the chemistry and biological measurements in the field in order to do that. We do this by combining our monitoring work with chemical and biological studies along with our modeling work. We actually embed the chemical and biological changes in the models to develop predictive capabilities, what the overall impact would be to an organism or to a system. And we now have the capability to do that. Next slide, or next point. Second point is that we have to combine the field observations and observe these changes and look at it over different time cycles, different long-term and short-term changes, and relate that to the changes, not just the acidification itself, but the other stresses, the changes in the temperature and the hypoxia, and be able to understand what the overall impacts to the ecosystem are by these combined impacts. That's really challenging, but is still very doable. We can study in the laboratory these individual impacts. We can then go out in the field and see where they actually are combined and see what the impacts are. Again, the approach is by combining our monitoring and modeling, we can provide real insights on the combined impacts of these combined stressors. Next point. And finally, what we know for certain now from studies that have been done both in the laboratory and in the field in some cases, that oyster larvae, pteropods, crab larvae in some cases, forams, they're all exhibiting impacts of ocean acidification right now. We can see these changes in the field. And as we take these long-term measurements of the biology, we will be able to see how they're changing in the field in the future, and then combine that with our models to make good predictions of what the ecosystem response will be. And again, the way we solve this is by continued collaborative field and laboratory studies combined with the modeling. I'll stop there and turn it over to Tessa.
Great, thank you. Unsharing your screen. Tessa, we have you on deck. Next, we'll hear from Dr. Tessa Hill from Bodega Marine Laboratory, University of California, Davis. Dr. Hill is a professor and chancellor's fellow at UC Davis in the Department of Earth and Planetary Sciences. She is resident at UC Davis Bodega Marine Lab, a research station on the Northern California coast. Her research interests include climate change, both past and present, and understanding this response of marine species to environmental perturbation. She is part of the Bodega Ocean Acidification Research, or BOR group at Bodega Marine Lab, which aims to understand the impact of OA on marine species. So today she's going to be talking about ocean acidification, what have we learned through observations in California. So Tessa, we see your presentation and we will turn it over to you.
Okay, great. Thank you so much, Haley, and thank you, Richard, for that excellent kickoff to the webinar. I turned off my video because my internet connection was kind of unstable, so you all are going to have to flag me down if this starts not working very well. We'll see. I was tasked with thinking about what we have learned already, and so I'm going to talk about that, but the first thing I'm going to do is just tell you sort of some things I want you to take home from our talks today. These are things you will hear over and over again, and in fact, several of them you just heard from Richard. You're going to see a lot of sort of repeating themes in these talks. We're dealing with a system that's tremendously variable, and so in planning a monitoring network, we have to keep that in mind. Partnerships have been real winners in paving the way to be able to understand and respond to this problem. And long-term studies of the ocean, really having our eyes and ears and hands in the water, are pretty essential to understanding today's changes and putting them in context. So the first part of this talk really focuses on what we've learned so far. And you saw this excellent figure already from Richard. This is the longest time series that we have in California based on measurements led by Francisco Chavez and colleagues at MBARI. And Richard did a wonderful job of walking through this, so you can all see the increase in carbon dioxide in the top panel and the decrease in pH in the bottom panel. And I'll just add to that we also have NOAA moorings offshore California that have existed for a shorter time period than this, but the data would overlay right on top of these. So we have a very consistent story about what's happening from the few long time series that we have in California. Another really nice element of this study and this paper is that it ties it to subsurface and biological impacts. And so the bottom two panels are what you already saw. So underway carbon dioxide measurements through time and a calculated pH through time. You just saw those in the previous slide. But then on top of those two, you see measured oxygen concentrations. These are measured at about 300 to 400 meters water depth, and measured sightings of jumbo squid. And so what you're seeing is both changes at the surface as well as chemical changes in the subsurface and a biological response to those changes. And so we need to be sort of keeping track of all of these things, and this research has really led the way in trying to capture all of those elements. Another thing that I think we've learned, which you also saw in Richard's talk, is that there's a tremendous amount of variability within this very complex system. And so what I'm showing you here is an image from a paper that we published with Richard in 2016. And it paints this picture of a highly variable system, a real natural laboratory to understand adaptation and pH variability along the coast. These are pH values in the surface in Washington, Oregon, and California. And we were able to paint this picture because we combined data from a NOAA study, from a NSF-supported study, and these data were led by Francis Chan, and by a study that we had done here at Bodega Marine Lab. And so by pulling together all of those data in partnership, we were able to get this sort of snapshot of how variable the coast can be. Why does that variability matter? Well, some great papers by Christy Croker and Francis Chan articulate very nicely that variability really matters to organisms. And so I'm going to quote Christy here because I've heard her explain this so well, that we often imagine that ocean acidification is like pulling up a blanket along the coast of impacts. And in fact, it's actually a patchwork quilt. And so if you're an organism along the shore, where you are on that quilt, which patch you're in really matters. Whether you're in a low pH patch that aligns with a high or low temperature patch or a high or low food availability patch, all that variability really impacts how the organisms will respond. And so any monitoring that we do to try to understand the California current, and more specifically the California coast, has to sort of take that variability into account. I'll also say that we have several examples now of ocean acidification responding to very short-term temporal variability. And the way we've been able to do this is through studies led by Simone Allen, Lori Juranic, and Catherine Davis, 3 individual studies that developed empirical relationships between temperature, salinity, dissolved oxygen, and carbonate chemistry. And so what that means is that we can take long time series where we've measured temperature, salinity, and oxygen, and we actually can reconstruct the carbonate chemistry piece. And so I'm just giving you one example here from offshore Bodega, where you're looking at aragonite saturation in the bottom data with the right-hand axis, and dissolved oxygen as the top data, and it's through time in 2014 and 2015, and the colors are showing you the density of that water. And what you're seeing there is in 2014, in spring of 2014, you're seeing these sort of low aragonite saturation state waters that were, sort of typical during the spring and summer offshore Bodega. And then the marine heat wave hit. And so for the remainder of 2014 and 2015 and into 2016, we had very warm waters along the California coast, and that really ameliorated the ocean acidification stress temporarily. And so these short-term events that we're starting to understand also influence how we wrap our arms around ocean acidification. Finally, I'll just say that one of the things we've learned is that we have to capture both the local and the global problem. And so in places with high nutrient input, we have an exacerbation of the ocean acidification signal. And even in the absence of nutrients, riverine inputs or terrestrial runoff sort of complicate the coastal signal of ocean acidification and can do that in multiple different directions. And so we have to really understand both local processes that might influence ocean acidification, as well as sort of coast-wide or regional or global processes. So I've been telling you things that I feel like we've learned over several years, and that is a good segue into sort of what we need next. And you're going to hear a lot about this from the next two speakers, but I'll just point out a few things that we know. A recent report just came out from the California OAH-relevant office, Indicators of Climate Change in California, and it covers some of these things we're talking about today, including that assessments of changes to the carbonate system rely on a handful of data records. And we really need to take that handful up to, an armload. And in fact, in this report, it states we need a robust integrated monitoring system for OA, hypoxia, and biological impacts. As Richard pointed out, a really exciting starting point of that is the growing IUS network, and in particular, a growing network of sensors that have been done in partnership between the Integrated Ocean Observing System, the NOAA OA office, and aquaculture hatcheries along the coast. And so because of this partnership, and because of the leadership from IUS and NOAA on this, we're actually really starting to build that network, which is exciting. And I'll zoom in here for a moment on just the California piece of that, with a real hat tip to Senkus and Scoos for being leaders in developing these data sets, supporting these data sets, etc. And another thing I'd like to point out is that the most recent of these to go in, oh, that arrow is pointing at the wrong location. The most recent one of these to go in is not where that arrow points, but is in fact down in Humboldt, so the next sensor's down. And this is exciting because it was a collaboration between Sea Grant, the California Ocean Protection Council, Humboldt State University, and Hog Island Oyster Company. This, we're able to actually see some exciting things from these partnerships developing and putting instruments in place that are developing and sort of leading a path for that. So another thing that I think we have learned and that we need is that co-locating these sensors, chemical, physical, and biological sensors together provides real insights that we need in this, a direct measure of biological impacts and a measure of how complex those impacts may be. Biological monitoring also provides sort of the real world connection and the ecological relevance to what we're observing in the ocean in terms of chemical and physical change. And I'll just point out that consider where we would be if we hadn't monitored biological impacts associated with El Nino or Pacific Decadal Oscillation. In fact, we would be nowhere because we actually understand the physical processes in large part because people made the biological observations first. And so we need these things to be done in partnership. Some examples of people doing this well, as Richard just pointed out, we now have pretty good evidence that a lot of things that live at the base of the food web, pteropods, foraminifera, krill, crab, and oyster larvae, all of these things are impacted. And these are things that we can monitor together. If we're out there monitoring the oceanography, we can also monitor the sort of base of the food web indicators at the same time. Another great step forward is the development of ocean climate change indicators. This is an example of a report that was developed by the Greater Farallones National Marine Sanctuary that developed a suite of biological indicators that can be used for climate and ocean acidification processes in Northern California. These were done through an expert judgment process. And now researchers who are headed out to monitor oceanographic components of these processes can pair that with recommended species that are likely to be indicators. And I'll just put a little plug in here that I'm aware that the Ocean Science Trust is also leading a project to develop a longer list of species that are likely to be sensitive to these processes that we can watch closely in the future. Another example of these paired biological and chemical monitoring systems is the work led by the Omegas team, which was a consortium of universities doing work together. This really combined laboratory and field observations with oceanographic sensors. And one of the exciting things about this is that it makes me think that our existing amazing resources on MPA monitoring, NIR sites, LTER sites, anywhere where we've invested a lot of time to monitor the biology is an excellent place to then be monitoring the chemistry and the physics of this process. I'm almost done here, but I did want to make a plug for the fact that people are involved in these impacts as well, and this is summarized so nicely by Julie Ekstrom and colleagues in this paper, where, just to walk you through this in case you haven't seen this before, the purple here in the ocean tells you sort of the years that ocean ecosystems will cross thresholds, and that's held up against information about the economy of a region and how vulnerable people in that region might be to the effects of ocean acidification based on the economic reliance on fisheries. And so really when it comes down to it is our vulnerability to ocean acidification is this sort of combination of all that oceanographic variability we've been talking about plus the economics of the region. And we really need to be able to sort of even further with greater granularity stitch together how the ocean is changing and how people will adapt and respond to those changes. So Richard told me I got to write a wish list, so I did. This is my wish list of needs and visions. You've heard all of these or most of these through the talk. We need to invest in monitoring from the surface to the subsurface. We still know relatively little about both. We need to really utilize and expand partnerships with local communities, tribes, aquaculture fisheries groups, regional and local managers and scientists, because those partnerships seem to be a way that we make a lot of progress. Co-locating biological and chemical physical monitoring, you've heard that several times. And I'll just add in there that then synthesizing what we know about the modeled progression of this problem and thresholds with what we're actually observing in the field. And finally, any way that we can build linkages between that mosaic that I showed you of oceanographic impacts and pull it right up to the human impacts along the coast, that's going to be a place that we make a lot of progress. I told you that I'd also finish with these take-homes, and so these, this is just to remind you with what we started with. This is a tremendously variable environment, and we have to capture that complexity in any monitoring network that we develop or build. Partnerships between many different types of groups are a really amazing way to maximize and leverage our understanding of this problem. And finally, just a final plug that trying to understand these combined impacts of chemical, physical, and biological change is really important, not just that we're documenting, but it enables us to respond to it on the human, economic, policy, and management side. So this information is tremendously valuable. Thank you so much.
Great. Thank you, Tessa. All right, thank you, Tessa. Sorry about that. Appreciate your presentation. So our next presentation, again, we're going to save questions until the end, so please feel free to chat those into us. Our next presenter is Sarah Braley. She is a climate change fellow at the California Ocean Protection Council. Sarah joined the OPC in February 2017 as a California State Sea Grant Fellow. She worked on climate change projects related to sea level rise, ocean acidification, hypoxia. She earned her master's degree in biology from California State University Fullerton and her BS in marine biology from the University of California, Los Angeles. For her master's thesis, Sarah studied the impacts of native Olympia oyster restoration on co-occurring eelgrass. Sarah has been leading efforts to assemble the West Coast Ocean Acidification Monitoring Inventory, working closely with West Coast states, and she's going to share progress of that inventory with us today. So Sarah, take it over. Feel free to share your screen.
All right, thanks. Can you all hear me okay?
Yes, there's a little bit of background noise, but I'll stop my audio.
Okay. All right, so thanks for that intro, Haley. I'm just going to share my screen here. Okay. No, all right, I'm not sure why this is. Okay. All right, so I'm going to be zooming out a bit to talk to you today about an ocean acidification and hypoxia cataloging effort that's been underway since late 2016. So this effort has been led by the West Coast Ocean Acidification and Monitoring Task Force, which is a joint effort of the Ocean Acidification Subcommittee of the Pacific Coast Collaborative, which is a governor-level partnership among West Coast states and British Columbia, and the Federal Interagency Working Group on Ocean Acidification. And the task force is made up of members from British Columbia, Alaska, Washington, Oregon, California, and federal partners. And the California team in particular is made up of Jen Phillips and myself from the Ocean Protection Council, Clarissa Anderson from the Southern California Coastal and Ocean Observing System, or SCOOS, Alex Harper from CENCOOS, that's the Central and Northern California IUS, which formerly we had Eric Bickle when he was with CENCOOS, and we also have Gretchen Hoffman from UC Santa Barbara. So specifically, the vision of the task force is to work towards improving ocean acidification and hypoxia monitoring capacity along the West Coast through jointly building an integrated monitoring network that is responsive to management needs. And so the task force will go about accomplishing this by completing an inventory of monitoring assets that are tracking physical, chemical, and biological indicators of ocean acidification and hypoxia. And then the second step is to assess gaps in this current monitoring network and prioritize filling those gaps. All right, I'm not going to spend too much time on this, but the background of the task force, it's a culmination and coordination of several different initiatives. Specifically, it builds off of recommendations from the West Coast Ocean Acidification and Hypoxia Science Panel that was released in 2016 to identify and improve gaps in the current monitoring network. And it accomplishes the Pacific Coast Collaborative initiative to design and implement coordinated, effective monitoring and research. And the task force became a joint state and federal task force after discussions with the Federal Interagency Working Group on ocean acidification, and a partnership was formed in 2016. Okay, so before I get into showing some of the details of the monitoring inventory, I wanted to spend some time in the beginning talking about how we put this inventory together. So while this is a regional effort to inventory monitoring efforts or assets along the West Coast, we started by tackling this huge endeavor by having individual jurisdictions curate their own inventories before we began the roll-up process of putting all of the inventories together for the entire West Coast. So though each process was slightly different, we all followed the same general process here. So first we started with a template that was developed over several months by the joint state, federal and state monitoring task force. And the task force decided on this template after really thinking about the types of information that they were most interested in collecting. The second step was distributing this template out through each of our jurisdictional monitoring networks. And so in California, that was largely led by Eric and Clarissa from those regional IUS associations. And we received a lot of follow-up. So if you contributed information, thank you so much. We're so appreciative of your time. Third step, there were one or two inventory leads from each jurisdiction who consolidated all of the responses submitted, and there was some level of translation involved in this step to get all of the information into one single source. Fourth, we had some initial quality control done at this point by those inventory leads, following up on entries that had missing or unclear information. And the fifth step, as we refined the information, the inventory leads were in very close coordination to be sure we were being consistent in how we were characterizing assets in the inventory, we developed definitions for each field and classification to be used by the rest of the jurisdictions. And then the sixth step, to better visualize this inventory, we uploaded it to ArcGIS Online. And Daniel Sun, who was the Sea Grant Fellow at the Oregon Department of Fish and Wildlife, created a web app for us to use to showcase the inventory and also used as a useful step in quality control. So that was the seventh step, was sending this web app out again to our monitoring networks to make sure that everyone was comfortable with the information that we had collected. And then the eighth and sort of final step is integrating all of the jurisdictions' inventories into one single West Coast wide inventory. And so where we're at right now is we have most of the states together. Alaska and British Columbia have gone through that previous step of quality control using the web app, and we're just finishing filling in some missing information now. Okay, so the next thing I wanted to talk about, which may be self-explanatory, but these are some of the basic criteria that we used to decide whether projects were included in the inventory or not. First and foremost, they had to measure at least one metric of ocean acidification, so pH, PCO2, dissolved inorganic carbon, or total alkalinity, or hypoxia, so dissolved oxygen. They also repeatedly measured assets to document changes at a site over time. So we didn't have any projects where only a single measurement was made at the site. They were all projects that were undertaken over time. And then this is a purely observational inventory. We did not include any projects which had experimental or laboratory-based components. And I do want to note there's some programs that we did include in the inventory that did not directly measure OAH directly, and those were largely biological data sets. So a lot of the biological sampling programs that we have in California didn't directly assess physical or chemical measures of ocean acidification or hypoxia, but we felt it was important to include those in the inventory to better understand the capacity of current monitoring networks to detect impacts of OA and hypoxia and demonstrate efforts that could be augmented to build monitoring capacity. All right, so some of the types of data that we collected, this is the template that we sent out. So we collected quite a bit of information on not only the types of data collected, but also more operational characteristics like funding and end date. So we collected basic project info and background information like project name, institutions involved, point of contact. We collected more information on the types of study, what type of asset are we looking at? Is it a mooring, a shoreside sensor, cruise station, glider, for example. We collected information on locations and depths, also temporal characteristics on when and how often measurements are made, which OA and hypoxia parameters were measured, and also what methods were used. Were the samples collected in situ or via water sample, and what instruments were used as well. We also looked at other water quality parameters like temperature and salinity and nutrients, and information on biological sampling. Funding information was also collected, such as the source, end date, and likelihood of future funding. Okay, so enough background. Let's look at the inventory. Can you all see my web app? Great, okay.
Yep, looks great.
Okay, so I'm gonna skip this. All right, so this is the web app that Daniel Sun created, and it has all of the current iteration of the monitoring inventory that goes all the way from, we have got some actually in Baja, Mexico, but we go all the way up to Alaska. I think we've got one point up here as well. I can't get up there. There they are. But yeah, so this monitoring inventory is if you remember from Tessa's slide of the IWG-OA data portal, it's substantially expanded from the version of what we currently have on data portals. So there's a lot more monitoring on here that hasn't been consolidated before. All right, so I'm just going to spend some time walking you through the web app. I think we'll be able to share this after if you'd like to browse it yourself. I'm going to mostly be focusing on California now, but feel free to look at the entire West Coast. So there's different layers here. I just want to point out. I'm not sure how to get rid of I got it. Okay, so we got point assets here, and those are monitoring assets that are represented by a fixed position in space throughout time. We also have sample tracks, and these are monitoring projects that are collecting measurements underway. And then we also have sample areas, which are projects that are using a systematic monitoring method within a specified area, and they weren't able to refine it down to a specific point. So we've included it as a sample area for now. All right, that's the layers. You can also turn on the legend to see what the color coding is about. I think I turned off all of the layers, so let me turn those back on. Right, so I just want to point out here, so for those point assets, you can see the dark green points are showing where projects are monitoring both ocean acidification and hypoxia. The light green points are sites that are monitoring just ocean acidification. These light purple points are showing projects that are monitoring hypoxia. And then there's a lot of dark purple points that are showing projects that aren't monitoring any measure of ocean acidification or hypoxia. And those mostly are sites that have extensive monitoring that were added, even though they don't monitor OA or hypoxia, but they've been monitoring long-term impacts, or they could be augmented to substantially increase OA monitoring capacity. Okay. We also have, since this was created primarily as a way to, for monitoring practitioners to visualize and verify their information, we set up a filter where people could filter by contact person or organization or project ID to find their information quickly. So here, if you can see them, are some of the sites that Francis Chan is listed as a point of contact for. And you can click on these project points to find out more information about what's going on. There's a link to find the data, what type of institution, what type of asset. We have the years that the data was collected and which parameters are monitored. Many other things you can take a look at here. So you can also find information on the assets by looking down in the attribute table, and there's an option. If you go into options, you can also download all of these data as a CSV file, so that you can look at it in Excel rather than spatially. All right, so I think I'm going to move on now. I do want to say, though, that this is not the final format that the inventory will be hosted in. It's also, we're still, like I said, adding more assets to this inventory, so it's not complete. But it's currently the best way to access the inventory at this time. All right, so let me go back to my presentation. All right, so if we look at all of the assets that we have in the monitoring inventory, it looks like we're doing a really fantastic job of monitoring OA along our coasts. But if we start applying a series of filters to the data, we get a better sense of how well we were actually monitoring. So if we remove the monitoring sites that are no longer active, we get a slightly restricted view. If we want to focus solely on the sites that are monitoring carbonate chemistry, we also see many projects drop out here. And our network becomes even smaller if we're only interested in observations that are collected continuously over time, meaning they have in situ instrumentation collecting high frequency data. So there's already obvious gaps in this view of places where we aren't capturing the full temporal variability of ocean conditions when we're only able to collect spot samples or infrequent measurements. But we could continue to filter this by using other characteristics like funding stability, depth, whether they're looking at surface or subsurface, and also data accessibility, how publicly available are these data sets, which would likely create even more gaps. And so these are the sorts of gaps that the Monitoring Task Force would be working on next to highlight and prioritize filling. I also want to go back to another point that both Richard and Tessa made about the importance of co-locating biological and physical chemical monitoring, which was also an important gap that the Monitoring Task Force recognized early on. So here are the monitoring projects that are monitoring both physical, chemical, and biological observations simultaneously. This might give a slightly skewed view because the majority of these are monitoring only low trophic level organisms such as phytoplankton and zooplankton. So these are the sites where long-term biological monitoring is taking place in California, which are not collecting or collecting very limited physical chemical observations. Many of these projects are monitoring higher level trophic levels. And so if we were to add more physical chemical monitoring to these sites, we would substantially expand our monitoring of ocean acidification and better track impacts on our ecosystems and organisms. This inventory also presents an opportunity for researchers across disciplines to assess what additional types of monitoring are occurring in close proximity that they may not have been aware of. So there are endless ways that you could filter the inventory using the information we have collected to identify gaps for a myriad of different management questions you're asking, which is how we hope the inventory will be used in the next phase of the task force work. So to wrap up here with next steps, like I said, the inventory is nearly complete. Alaska and British Columbia recently finished their public review of the original inventory they put together, and we're working on filling some of the missing information in there. At the same time, the task force is working with partners to host this information online through existing data portals so that the inventory can be easily accessed and downloaded. And in winter 2019, the task force will bring together state and federal policymakers and scientists to evaluate the inventory to identify and fill critical gaps in monitoring along the West Coast. So I think with that, this is a good place for me to stop and turn it over to Karin, who will talk more about how we can use this inventory more.
Great, thank you so much. I appreciate that. That's a huge effort coming together, and it's great to see the progress. So next, we're going to pass it over to our last presenter, Dr. Karin Braley. She is a Marine Resources Program Manager at the Oregon Department of Fish and Wildlife. She's been with ODF&W for 10 years. Her work is grounded in both fishery and ecosystem issues, and ocean acidification has become a focal point for Karin's work over the past 5 years. Dr. Braley provides strategic leadership for the state of Oregon on West Coast ocean acidification and hypoxia policy issues, working closely with the Pacific Coast Collaborative and the West Coast states. Dr. Braley has a PhD in biology with a focus on marine invertebrate physiology and ecology from Stanford University Hopkins Marine Station. So with that, I will pass it on to her. And looks like we can see your presentation, so here we go.
Wonderful, and you can hear me?
Sounds good. Thank you.
Good. Great. Well, it's an honor to be part of this panel and part of the next steps for the Science Task Force and for California as well as the region. And I'm coming to this panel and to this webinar with a couple of perspectives, and just for full transparency, want to reveal those right up front. And Haley already introduced some of them, one of which is that I'm a biologist. And so all of the chemistry and physical processes that have been described in just wonderful detail are great. I come to this being interested in the animals and the animal response. And Tessa spoke to this a bit and emphasized that a lot of us who are in the management realm and the decision makers that we work for and work with really care about the biological impacts, both to the species that we rely on and the related human impacts that are cultural and economic impacts. And so it's really kind of digging deep back to that, that core human interest in the biology and making that up front and center in terms of the science that we bring to the table is critical for getting our community broader than it currently is. And I'm delighted to see that this webinar has over 100 folks tuning in to help think about what the solutions might be. And I challenge not only the task force members, the 8 of you, but everybody on this webinar and those in your networks to think about how you as an individual and you as an individual within your institution can help be part of the solution. And I think that's the spirit with which we're all in this together today. And I borrowed this just absolutely gorgeous picture of a pteropod from Nina Bednarsik, who, along with her colleagues, has championed the pteropod as kind of the face of ocean acidification, the species that can't speak for themselves. And so with this, I'll kind of move into describing the management and policy framework that has developed over the last few years. My subtitle here, Getting to Work, is really a message to myself, and it's something I repeat to myself almost on a daily basis. Okay, today I'm going to come up with the solution. I'm going to move that next step forward to try and make this a better situation for us all. So as Haley mentioned, I have been working with partners on the Pacific Coast Collaborative, and particularly the Ocean Acidification Subcommittee, which is a team from British Columbia, Washington, Oregon, and California, policy advisors, staff of agencies, folks from different areas that are all advising the Pacific Coast Collaborative at the governor and premier level on ocean acidification policy development. And early on, this has been something that has focused on kind of task-oriented approaches to increasing the dialogue between science and managers. One of the first things that we worked jointly on was something that was started by California and the Ocean Protection Council and the Ocean Science Trust, which was standing up a science panel on ocean acidification and hypoxia. And that quickly became a coast-wide effort that started in 2013, ended in 2016. It's already been mentioned by at least one, if not all 3 of the previous panelists. But the reason why this science panel is so meaningful is that it really kind of created a shift in the understanding of where we were with the science and what we needed to do in order to make progress. That panel created a roadmap, but most importantly, from my perspective, it really challenged scientists and managers to work together to develop solutions. Of the 6 major findings from the panel, there were 2 that I looked to in particular as being very important. Everything they said was of great importance, of course, but the second finding here, the very transparent admission and challenge to us that ocean acidification impacts that we were seeing stemmed from a causal factor of carbon emissions was critical. And this very transparent, very blatant finding has been incredibly useful in policy discussions within Oregon. Secondly, and really to the point for today's webinar topic, is that scientific understanding expands the options that are available to us. And right now, management, as a manager, I can tell you that we feel like the management options are not voluminous. And so we are seeking to create options. We are seeking to create understanding so that we can see a more diverse set of options than what we currently have. The science panel also spoke specifically to the monitoring network and criteria associated with it. And again, important for today's discussion, number one, supporting the needs of decision makers, looking for those opportunities where managers make decisions, and having a network that informs those decisions is key. The second, that the network measure physical, chemical, and biological variables is also very important for the reasons that Richard and Tessa went through beautifully in their slides. Number three, also very important to managers, building on what is already in place. And so instead of recreating the wheel, spending additional funds on redundant systems, we need to take what we have that's good, augment it, and manage it to the best of our ability to give us the best information that we can get. And then fourth, of course, that monitoring network needs to enhance what's happening in the monitoring community. So one of the second things that Sarah talked about a bit was that the PCC invested in this relationship with the Interagency Working Group on OA, the federal committee that was established through the Congressional FORAM Act in 2009, and Libby Jewett, Shallon Bush, and Richard Feely, just to name a few from the NOAA family, and who are all associated with the Ocean Acidification program there, have been very instrumental in establishing this joint task force to conduct a regional monitoring inventory that Sarah showed you and is in just the final stages of becoming a final product, a final living dynamic product. And she already described that this is a catalog of projects rather than a data set or a set of data sets, but it provides discoverability of those projects and not only includes the Pacific Coast Collaborative 4 jurisdictions from British Columbia to California, but also includes the monitoring that's occurring in the state of Alaska. Next steps, we are currently on target to complete our final QA/QC this summer, and Sarah has been just fantastic bringing this project towards completion this summer. We are still working on different options for hosting the inventory, but all of the partners involved see that accessibility to this inventory is absolutely essential. And we hope to host it in a place where there's a stable, single source for the information, but that it is mirrored all over the place and to the degree that other institutions want to mirror it and link into it. We hope to have those options finalized and have it hosted this fall, but as Sarah mentioned, you can already get into the monitoring inventory. It's not transparent, but we can get you information so that you can see it if you haven't already. And then we get into the gaps analysis. The purpose of having an inventory is not just to show, hey, this is what we're doing, but rather to use it to inform how we can do it better. And so the gaps analysis is essential and so critical to our next step. As Sarah mentioned, we're planning on doing this in the winter. And one of the things that I hope will come from not only the California Science Task Force, but from the other jurisdictions is ideas on what those scientific priorities are for the gaps analysis. And that in these individual jurisdictions, we can build some ideas, bring to the table in a regional gaps analysis process, and come up with the common ground approach for the large scale, the basin scale regional priorities for strategic investment. And that's the take home. We want this monitoring network to persist. We want it to be as maximally informative as possible. And I want to stress that even though it's not finished now, it's already been incredibly useful and has been the inspiration for a number of projects, one of which Tessa described to you at Hog Island. So in addition to the monitoring network, I wanted to kind of scale even farther up to share with you that we have a policy framework into which science recommendations can fit beautifully. And this is something that the PCC group has been working on for a couple of years now. It's been called the International Alliance to Combat Ocean Acidification, or the OA Alliance for short. And it is a group, voluntary group of governments and affiliate institutions that was stood up by the West Coast jurisdictions as well as partners, Ocean Conservancy and the nations of Chile and France were all founding members. And this group developed a call to action for governments that would join. And the primary stipulation is, come join us. And if you do, as a member, we expect that you will commit to making an ocean acidification and hypoxia action plan. Building an OAH or OA action plan, as everyone on this webinar knows, is not a simple thing. Finding the right solutions for each place, each jurisdiction is difficult. But the call to action for the OA Alliance includes 5 major areas where actions might be developed for each jurisdiction developing an action plan. And right up at the top is advancing scientific understanding. Achieving success with number 1, of course, supports actions in the remainder of those 4 areas and is critical to developing rationale and building convincing arguments about taking costly steps to address ocean acidification with resilience measures, adaptation measures, and mitigation measures. Now, in terms of our West Coast jurisdictions, the state of Washington put in place their first OA action plan in 2012 through the activity of the Blue Ribbon Panel, and just recently updated that, that report with an addendum that was released in December of 2017. Their work has been leading not only themselves, but the West Coast states as well. And the state of California is on target to finalize an OA action plan this year. And Oregon is close on California's heels, trying to develop a draft this year and a final plan next year. Now, all of these plans that are in place or soon to be in place are part of a greater commitment that the OA Alliance has made to the world, which is by next year we have 15 action plans in place. And you can see some of the international and national partners in the logos at the bottom. It ranges from cities to counties to states to nations to tribal governments, and brings together that rich community of all types of government and affiliate institutions to the table. So here's the juncture. We have the opportunity. I feel like, as I said, getting to work, I say that just about every day. Okay, we did all of that yesterday. What do we do next? And the opportunity that we have now is that we have science alignment that has come from a lot of sources, but what I focused on today is the West Coast OAH Science Panel. We have established science and decision maker dialogue that has been in place for multiple years now. We have this almost complete inventory of OAH monitoring for the West Coast. this is going to be integrated not only and accessible on the web, but also will become part of the international monitoring inventories and data portals. And I've just described an international policy framework for action and some of the rich partnerships that have developed on the West Coast. We also kind of independently within jurisdictions have built up education and engagement with decision makers to be poised to actually take on the difficult decisions that need to come in terms of managing carbon, in terms of responding to ocean acidification. So we have all of these things going for us. All of these things are in place or almost in place. And what we need to do now is, for the monitoring network specifically, is make sure that it is supporting all of this framework and all of these decisions that need to be made. We need to identify the most strategic investments we can, because I'll tell you, my budget is not expanding right now. My budget is shrinking, if anything, relative to costs. And so the money that I have to spend as a manager is limited. The money I can seek through grants is limited. What do we do that will give us the best information with that limited amount of money? So again, I'll return to the biological impacts piece of this. And our knowledge about ocean acidification, as Tessa mentioned, came from watching the biology. It was the oyster industry that started failing in their ability to raise young oysters that really brought this issue to the attention of scientists and managers and decision makers alike. And in the Pacific Northwest, this is hundreds of millions of dollars of industry that serves the coastal rural communities of these two states, Oregon and Washington, but also is important in rural communities in California and British Columbia. But it's not just aquacultured animals, it's wild species that are of critical importance, and impacts on these other species speaks volumes to decision makers. Pink shrimp, Dungeness crab, Pacific cod, which is part of, for example, Oregon's distant water fleet activities. Salmon, these are iconic species for the North Pacific. And if we identify biological impacts that affect these species, then we will be in the same place with commercial fisheries as we were 10 years ago when economic impacts were facing the oyster aquaculture industry. And it's not just commercial fishing either. It's not just commercial endeavors. Recreational fishing in Oregon is at least as valuable as commercial fishing in terms of driving coastal economies. And so legislators, governors, they listen when these kinds of impacts are discovered and identified, especially if we have solutions that we can offer to help minimize those impacts. So gaps in investments. I've already mentioned that I'm a biologist. That's my bias. I'm interested in the biology. I love hearing about the oceanographic process and learning about that. But what I have in my hands as a manager is a lot of fishery-dependent data and not a lot of fishery-independent data, and long-term data sets are few and far between. And so as the California Science Task Force works towards identifying gaps for California, I'm hoping to see some really solid work, particularly on how to monitor fishery-independent impacts on biology. What is it that we monitor and how? In terms of oceanographic gaps, I look at this as having kind of two different areas of interest. One is local priorities, and the other is regional priorities. And I'll describe these a bit more as I move on. And for local priorities, a really great example is one where we've had very recent success. Francis Chan, who's on the California Science Task Force, was involved in this project. And Tillamook Bay is a really important estuary in Oregon. It's our second largest estuary. It's one of 4 that produces oysters for aquaculture. And there's no long-term monitoring that exists in Tillamook Bay as of right now. The US EPA and OSU researchers have successfully put together and are about halfway through a 2-year grant to identify water sources, pollutant sources, water quality, hydrography of Tillamook Bay. And they're putting together a really great project that is going to be fundamental for understanding how Tillamook Bay works. But in order for us to then document long-term trends, we need some instrumentation there. And this rationale, along with the progress made on the inventory and the West Coast collaborations and partnership, helped us put together a proposal to fund a long-term instrumentation package for the Tillamook Estuary Partnership, which is based in Tillamook Bay. And we're building their capacity over a 2-year grant to understand how to use the instruments, how to get the most out of the instruments, and then put together some work to actually continue this in the long term and build that local capacity there. So that's an idea of a local priority, and the point being that those types of projects I anticipate will happen because of, or at least in part of, because of the inventory. The regional priorities are more difficult for managers to get our heads around and to really make progress on. And that's where I see scientific debate and discussion and development of priorities being absolutely essential. And it really comes down to some comparisons that both Richard and Tessa already shared, which is we have observational monitoring happening, we have modeling happening, and we need to figure out how we as local managers can invest in monitoring that will feed into these much larger basin scale efforts and inform these models so that they are as rigorous as the scientists feel like they need to be in order to inform local priorities. So that's an iterative process that I see as being very essential for minimizing the amount of money, the amount of time, the number of staff that we need in order to get the observations, but then also maximize the scientific value and therefore the political value of monitoring. So for the task force, I think I've already said this a couple of different ways, but I'll say it one last time, which is that any actions that we take as managers have to outweigh the costs. And that's not a dollar for dollar comparison, but we need to be able to have the scientific justification that actions that we take that are going to be costly culturally, economically, are worth the cost up front of us putting something in place. And we need some science to show us where those benefits are. Task force scientists, what I think I, as an individual, think that we need from you and for you to wrestle with is which sites do we need more observations from? What can be modeled? How can we minimize the costly field observations? And when you are measuring, when you're actually taking observations in the field, what are the least cost options? And Tessa mentioned reconstruction of aragonite saturation state from temperature and salinity information. Options like that are absolutely doable. We have such a rich history of temperature and salinity measurements on the West Coast, but not so much in terms of 2 or 3 ocean acidification parameters at the same place at the same time. We also need to know which species are really at risk from a population level sense. And pteropods clearly are at risk. and that implies that others that forage on them, for example, are also at a population level risk. But more information about that and how monitoring can help us inform that is critical. And then a little bit more far afield is what resilience building actions can be informed by a monitoring network. And as a manager, I participate in decision-making processes that are not specific to ocean acidification, but they are specific to building resilience in the system. And yet there are often problems that face me in the same way that face consumers, for example. You know, is it better to buy something that has plastic wrap on it, or is it better to buy something out of metal that takes more energy to make it in terms of overall footprint on human resources? In the same way, when I'm looking at resilience actions, if I try and make a decision about a fishery that builds a little bit more resilience in that species, is that meaningful at the population level? Is ocean acidification related to that? And what's the cost-benefit trade-off? So the promise from me as a manager to the scientists is that we can help identify the species that are most important. And we are seeking more definition about what actions we can take, and we're trying to create places where we can take additional actions in a regulatory framework, in a state management and policy framework. And so that's my commitment to you, the scientists, that's what we bring to the table. And our partnership in finding solutions is essential to us both being successful. So I am done. I'll end with the inventory. And again, I'm grateful to be part of this webinar today and have a chance to talk with you all about this important set of issues.
Thank you, Karen. I appreciate all the presenters for presenting today. And now we're going to transition into our discussion. And I have a slide to share with some discussion questions. And this is really to kind of stimulate discussion among the task force, and this body is helping to inform future investments and priorities in California and beyond. So we're hoping this is a start to the conversation, and then we will open it up, hopefully at the last 10 minutes, to questions from the public. And I do see a few chatted in here, so I hope we can get to those as well. So I'm gonna share my screen. Let's see. Just share screen. Alrighty. Okay, and Dick, I'm gonna transition over to you to lead this discussion. Our first question is up there on the screen.
Well, thank you very much. And this was wonderful presentations by all the panelists. I thank you so much for your effort to do that. This will be a discussion with all the task force. So those of us who have been on the task force, please feel free to chime in at this stage. We really want to take the advantage of the discussion that we've had so far and kind of reach out and think about what this means for the future of our observing system. So the first question we have is, what is the major gaps in our present understanding of the processes on the West Coast, both at the fine scale that Karen was talking about and at the larger scale? What processes do we need a better understanding in, from the perspective of the chemistry and the biology together? Tessa, you want to say a few words about that?
Sure. I wasn't sure if you wanted other members of the task force to think about this, but yeah, I mean, I think we have a couple of gaps in terms of thinking about these things all together. One is thinking about sort of the multi-stressor standpoint of how organisms respond to change. And so there's a lot of different things changing all at once, and OA has to be seen in that context. And when we're thinking about organismal impacts, of course, we're thinking about a whole range of things, including behavior, growth, physiology, populations, mortality, etc. And sort of jumping off of that, we should be thinking about thresholds. Is there a place where we can predict through modeling or observe or both, where we will cross some sort of threshold in terms of species? And then I guess the other sort of gaps that we have are connecting those things to economic impacts and local scale solutions like were just referred to in Karin's talk. So are there things that we can do at sort of the watershed scale or the estuary scale that allow us to sort of push on some levers to protect things locally?
Well, do you or any other of the members of the task force have a vision about how we can address these combined stressors and their appropriate thresholds with a combination of observations and modeling? How would we go about doing that? And are we in a position now that we can do that? Francis, what do you think?
Well, I think that's a very important question. I would say I would think of it that we have two answers. One is, of course, we're really not there yet, and it's going to take some real investment and real commitment to make that happen. And looking at some of the questions that are on the chat, people are asking, how come we don't know more about things like behavioral impacts, for example, in the U.S. And I think it's important to note that, we're trying to do global change science in the U.S. on a very, very small budget. The federal OA program, I think this peak might have been $20 million. You can correct me, of course, Dick. But we're trying to address this very, very big global challenge on a very, really kind of meager amounts of investment. So we need to change that. But the other part, I think, for us as a task force, a scientist task force, is that given what we know today, what can we say? Are we still stuck at a place where there is nothing useful to be said? Or can we say, this is the state of the science, and given what we know today, these are the likely thresholds, or these are the likely well-supported decisions that can be made given, again, the state of the science. So two ways to go at it, I think.
It occurs to me that as we get a better understanding of how the biological systems respond to the thresholds, that allows us then to put that information directly into models to tell us where that response will be most acute, right? And where we might expect the major impacts to occur, or on the other side of the coin, where we might expect to see the best refugia. So it allows us to develop strategies based on our understanding of those thresholds. Do you think that we're in a position now to link up the threshold information with the models to get that sort of overall perspective?
Let me defer to maybe someone like Steve, who's been active in or Ali, who's been active in thinking about thresholds at the state level.
So, Dick, I think that's actually something that we're working on. It's in fact going to be the focus of the next task force webinar, probably to take place in an August timeframe, because I think you were exactly right. It is exactly one of the questions that we need to have an answer. All the monitoring is of lesser value if we can't define critical interpretational thresholds of how close we are, how rapidly we're moving towards those. But I think what I'd like to do is maybe, you addressed the question of, what are the major gaps in our understanding? And I think, Dick, in your talk, you actually hit upon it quite a bit, which is the interplay between modeling and monitoring. And I think to address a lot of the management questions, we're looking at essentially two types of models that are going on. Modeling number one is essentially what I call causal modeling. It's looking at processes to determine how much the local inputs, the local carbon inputs, the local nutrient inputs are affecting things, and whether or not, from a management perspective, their alteration or reduction would have a meaningful effect. The second thing we're doing is that a lot of what we're focused on is not the conditions at present, but how quickly we're going to get to conditions that are bad. And again, there's modeling that's going on to address that. I think the critical gap, focused on your question, is how tightly are we tying the modeling and the monitoring together so that those models are being well built and well affirmed that they are working now, and then it's a continuing way of validating those models as we move forward into the future.
Do you feel, Steve, that we're moving in that direction fast enough? Are we linking the biology and the chemistry such that the modelers can use that information in a time frame that's going to be useful, or are we looking 10 years down the road, or do you think we can do this in the next 1 or 2 years?
I think that's exactly the question that the task force has been asked to provide input on, so I'll speak only for one of us. I think we are absolutely on that path. I'm just seeing how far and how fast the modeling is moving forward. I'm seeing simultaneous projects to develop the thresholds that you talk about. I think that's all going to come together within that 2-year time frame that you just talked about.
Excellent. Let's go on to the...
Can I add one thing there, Richard?
Sure.
This is Tessa. I think I really like that vision and I endorse it. And I actually think what will hold you back is biological information on individual species. That is the gap there.
Yeah, this is Shalyn. I guess I'll chime in and build off of what Tessa said, is that, you know, we have a good amount of information on a few species about their sensitivity in lab settings, but understanding the balance of pressures in terms of predator-prey interactions, competitive interactions versus forcing from other environmental stressors like warming just creates a pretty complex picture to be able to attribute change in biology to OA.
So what's your solution for that, Shalyn? Do you have a way forward that we should follow?
Well, I think, you know, part of it is the modeling that you're talking about. Doing similar, so I know your lab has looked a lot about attributing CO2 change to carbon emissions. And you could see a similar track of attributing change in populations to these various environmental drivers to allow you to tease out signals. And, you know, there's a really big issue of signal and noise in a lot of the time series.
Anybody else have any thoughts?
Well, Dick, let me amplify what Tessa and Shalyn both said, because I absolutely agree. Well, I gave you a very positive answer. I think we're on the right track, and you know, the 2-year time frame to have answers that will be there, or I'm confident in. But those will be very, I guess I'll call them shallow answers. Those will be decisions that will be based upon biology at the level of the individual species. And even at the level of individual species, it will be at the level of the response at a physiological level, not necessarily even at a population level. I think that one of the things we've heard from every one of the speakers today is that the biology is key, and we're going to need to move from those single species physiological responses towards the interactive and population level responses. So point of emphasis there. I think we're all in agreement on that.
This is Jim Barry. I'd agree with a lot of what was just said, and it would be wonderful to have a coarse understanding of projecting success in particular populations with a future of environmental or ocean change. But the complexities that are going to start to show up outside of physiological responses alone are both the physiological sensitivities of different life stages that might or might not represent bottlenecks for population demographic processes. And perhaps even more important are those interactions that I think either Shalyn or someone else mentioned, that species interactions, particularly those where strong indirect effects may overwhelm the direct physiological responses, may not be apparent to us owing to our lack of understanding of how those indirect interactions are going to play out yet. But at the same time, I'd say that Steve's right. We're going to have a coarse understanding based on the literature that's developing already of a first order estimate of what those thresholds may be.
All right, let's go on to the next question.
Can I chime in real fast? This is
Go ahead.
Levin. I want to endorse this need to understand OA in a multiple stressor context, and how to approach this probably means that we have to throw all of our tools at the problem in combination, and I think we could make greater use of things like geochemical proxies that allow us to better understand what combined stressors organisms are exposed to. We can probably, from otoliths or shells or statoliths, we can look at oxygen, temperature, salinity, and other exposures all at the same time. We tend not to do that. But combining with use of natural gradients, use of laboratory studies, and use of modeling, I think we have to put it all together. Okay, thanks.
I agree. Thank you. Let's go on to the next question. We had a wonderful presentation by Sarah, and she did a great job. The question that comes to my mind, do we see major gaps in our observational network as it exists right now? And what are those gaps that we just really have to fulfill in the near term, both from the chemistry and from the biology? Does anybody have any strong feelings that there's obvious things we really need to get at and make sure we improve in the near term? Tessa, you want to say something about that?
Sure. I mean, this sort of comes back to what several of our presentations hit on. I'd say at the surface, our monitoring is not dense enough along the shore. We're really limited at the subsurface. We're really limited in sites where biological and chemical and physical processes are being studied together. And connecting all of those back to sort of impacts and importance to human communities along the coast is also fairly critical for a network like this, for the, to justify it and to get, useful information that managers can use and policymakers can use.
Yeah, I wanted to, go ahead.
I'd like to build off that. This is Shallon. The concept of pairing of chemical and biological processes, I think, is really important. And I'll just kind of riffing off of some discussions that are going on in the state of Alaska. It's not necessarily always enough to add biology measurements onto a chemistry cruise. If we are measuring the biology when the key processes aren't happening, we might be missing the boat. So maybe we need to start adding chemistry measurements to biological measurements when we know the critical recruitment processes are happening, for example. So that matching needs to be done in a way that's careful so that both processes are captured at critical times.
I think that's a very good, important point that you bring up, Shallon. I'm glad you stated that. Connecting up the biological processes with the chemical processes is really critical there. And you're right, we should be taking advantage of biological experiments in a way that we add the chemistry to them. I think that's a really good suggestion. I also feel strongly that we need to be real clear about some things that we've said earlier today, to need to emphasize. One of the issues of taking temperature, salinity, and oxygen and using those kind of information to get long-term changes take place, you got to be a little careful about that. That's good maybe for 4 or 5 years, and then you have to reset the clock because that anthropogenic CO2 is changing those algorithms. And so you have to have the observational systems in place on a regular basis so you can adjust the algorithms that you're developing and bring them up to date, if you will. And then you can do that testing from the observation to see how well the algorithms are working. So keep that in mind. Second point I wanted to make is that while we have a large amount of surface observations and the number of observations we're doing in the subsurface is quite diminished compared to those surface observations on the existing observing systems that we have. So we need to give a lot of thought to what we need in the subsurface and how that will provide us with the critical information that we need in order to provide the validation of the models. One of the things that did come out of this discussion is that there's lots of gaps in our observing system. And if we integrate the observations and models correctly, we can use the models to fill in those gaps. And we really need to think about what's the best ways to do that. Anybody else want to chime in?
Yeah, this is Karin. I just want to add another note about biological long-term data sets. And one of the things that we haven't done a great job of in Oregon, and this isn't the same story everywhere, is taking advantage of maybe citizen scientists or at least non-governmental science that's happening, and adding that to our ranks in terms of our ability to look at biological trends. And the biological impacts, as I've already said, as everybody said, is of so much importance, and we just don't have those long-term monitoring data sets out there, and we don't really know which species are the best ones to continue and to replicate along the shore. And so I think in combination with, integrating from lab experiments and models and observations, I think we need to think about, for biological impacts, we need to think about establishing a monitoring system, which is species and protocols that can be implemented by non-government scientists as well as government scientists, whether it's NOAA or ODSW or academics. And I think that is the only way that we're going to get the kind of information along this large spatial scale that we're going to need to understand those biological trends.
Okay, let's move along to the next question quickly. Anybody have any strong feelings about what new chemical and biological measurements we need to make, and where should they be located? Is there recent progress on the measurements of chemistry and biology that should be included in our observing system?
This is Lisa. Just to comment that it's become very clear that food availability is super important to how organisms respond to stress. So thinking about measurements that convey food availability, whether it's chlorophyll A or particle fluxes or something else, might be important.
Good point. Anybody want to add anything to that? Okay, let's go to the next question. How can we better coordinate our observational network? What gaps in our coordination do we need to fill? Karin, do you have some ideas about that?
I think that what we have demonstrated over the last several years is that when we all come to the same table and we share and pool our resources, that we come up with a much better picture of what is actually happening. And it takes work and it takes effort, but the outcome is so incredibly worthwhile. And I think the inventory is just one example of that. The Tillamook Bay pilot project that I described is another example. The Hog Island project is another example. We're hearing about them through academic collaborations and others. And so I think this regional discussion that we're having will develop the commonalities and the common approaches that we need to compare apples to apples across this region, and that the benefits of that will just kind of be a positive feedback loop. And so it's just kind of words of encouragement from me to keep doing this kind of thing, where we are talking across state and international lines to talk about what is really happening and what is important for us to track moving forward.
Good, excellent. Our last question is, how can we better utilize our models and observations, and how can we link them better? Is there a better strategy that we can have for tying the models and observations together? What would be your suggestions on that? Francis, what do you think?
Well, I was just thinking how, there are issues about data access that's pervasive in our observational network, and we can absolutely do a better job on that. But I think there's something that's similar on the modeling side, because I've had a lot of people want to get at the modeling data because we just don't know. There are places we've never taken a measurement, and we can't begin to guess what exposure might be like. So having the ability to access the model outputs from really different groups, I think that would actually help us to, I can poke around and compare my observations to their output, so that over time you gain confidence about where those models do really well and where they have a lot of trouble. So I think that confidence building is really important.
I couldn't agree with you more, both from the point of view of the access to the data. We need to improve that a lot. There is, inventory is just the first start on that. We need to have a mechanism that we can all get our hands on the data that we need to fulfill our tasks, and linking the models together with the data in a common location and a common website, so that we can test our ideas and work together with the modelers to test those ideas in a way there's good observational capabilities. I think that would be a major point forward in moving forward.
Some folks have a hard stop at 3, and can we move on to some of the public comments, unless there's a question? Please. So there's a question from Nina Bednarsik, and we're going to try and unmute you, Nina, so we will open up the lines to you. So yeah, have a chat now. It should be open. Nina, you might have to unmute yourself. Okay, no comment from, let's see. All right, Nina, try speaking again. Hello. Okay, let me see. Otherwise, we have a question from some other folks. Let's see. She can hear us, but we can't hear her.
Oh. Yeah, I don't know what to do. Let me try to mute it and unmute it.
So there's a question about hypoxia. So what are the current status or future plans of hypoxia forecasting on the West Coast? And are there examples of actions managers can take if they had seasonal to decadal hypoxia forecasts? I'll open that up to the group.
I'll just let you ask the question.
This is Karen, and I see that question, and that's a hard one for me to address. But just in this last year, we had a major hypoxia event occur off the coast of Washington, and during that same time, halibut surveys were ongoing for stock assessment purposes. And the result of the sampling occurring during a hypoxia event that was more or less obscured at that point led to a really low stock assessment result and constraints on fisheries. And so understanding when these hypoxia forecasts are happening and how that relates to management processes like stock assessments helps us understand what's happening and how we might make decisions differently to accommodate for those physical aspects. So it's a little bit different than maybe 20 years down the road what hypoxia looks like, but it is very relevant to management today.
Within the JScope effort led by Sam Seidlecki and others, there is an effort to do hypoxia forecasting out about 6 months or so for the state of Washington. I would be recommending that we extend that effort to a larger area. And the connection between hypoxia and acidification is really important, because the hypoxia intensifies the acidification by several fold. So we need to really develop those relationships and understand the underlying processes and how they connect in terms of the combined stressors.
Thank you. So we have another question, and this might be for you, Steve, but given the wide spatial variance observed in coastal areas, it seems like an important group to be working with would be land use managers who have the ability to address local regional sources of nutrients and carbon. To what extent is insight being communicated to them, and what guidance would you offer? And Ali, feel free to jump in for this as well.
So the answer is they're very aware, and there's been a lot of cooperation towards developing loading measurements from stormwater runoff, from wastewater outfalls, even from atmospheric deposition. Now the question is, how much effect does that have? And the modeling that Jim McWilliams is leading is focused directly on that question. Managers at both the regulated and the regulator level are very much in tune with this question. Some of the monitoring that you get to see in the inventory, and we'll see again more so in the inventory in the future, are moorings that they're putting out to help with validation of those models. So it's an excellent question, but I think it's one that they are very much in tune with. And Haley, you couldn't get Nina on the line, so she was right down the hall from me doing it separately, and she is sitting in my chair right now. If you want to, when she's ready, she can ask her questions.
Perfect. Go for it.
So I'd just like to comment on a few stuff that I heard comments being made today, and just in terms of like closing the gaps and maybe trying to provide some of the solutions. So here are my 2 cents. I think we have very little understanding on how the duration and frequency impacts. So how this variability impacts biological responses. And the second major is multiple stressors, right? So food availability, temperature, as Lisa mentioned before. And so, for example, one of our studies really showed how the temperature can be a mitigation against ocean acidification. So having really from the start on compilation of different stressors put together to see if one can alleviate the other, I think it's really crucial. So in terms of how to go about some of these problems, really complex problems, really, I think we have to try to really put biology into every time that the biological measurements are being taken. And maybe I've heard before that maybe biology should be measured just during the major recruitment processes. But I have a feeling that we should really be measuring it all across. So, because one of our major problems are the reference levels. So really some sort of comparison, the baseline is missing. And if we are just measuring the responses during the major bioprocesses, then we are missing out on those. I would also recommend that we are trying to measure the biological responses on event scales. So synoptic type of surveys combined with shorter time scales, and then get the biological responses measured at those scales as well. So really trying to cross different temporal scales, because we have no matching between the variability and biological responses in situ. And then on the thresholds, we have now been doing some synthesis work, and turns out that this is actually a really good way how to go about the thresholds. And so I would encourage maybe not just generate the new data, but also go about the synthesis, because it does provide a lot of an additional knowledge and understanding, and especially when it comes to delineating thresholds that are very difficult to be generated in the experimental conditions. So here, my 2.
Thank you very much, Nina.
Sure.
Anybody want to respond? All right, Haley, is there other questions?
There are some other questions, but we are over in time, so I wanted to be respectful of people's time. So if there's any, we will document all the questions that people have provided and make those available to the task force and anyone that was on this webinar on the westcoastoah.org website following this webinar. If there's any final closing thoughts, Dick, I will turn that to you, but I wanted to thank all the panelists for their presentations today, all the task force members, the Ocean Protection Council for funding this, and everyone who stayed on the line for 2 hours. So I appreciate everyone. And Dick, I'll send it off to you for some closing thoughts.
Well, I was really appreciative of the comments and the discussion today. I think it was very illuminating. We will have our task force meeting in Oakland, face-to-face meeting in Oakland on June 11th and 12th, and we will take all these comments and discussions as we consider the next steps. And so this has been extremely valuable. I hope that all of you that are out there, if you have the questions, feel free to contact Hallie with those questions, and she'll distribute it to each one of us. And I just hope that this is just the start of a series of webinars that we are planning on these topics and others. And one of them will be on the modeling, so we will be able to connect up with that. And another one will be on the thresholds, and we will be able to follow up on that as well. So I really think that this is a great start today, and I look forward to continuing to the discussion and adding to the dialogue. Thank you very much.
Thank you, everyone. And with that, we will adjourn this webinar. And thank you so much. Everyone have a great day. Or if you're in Poland like Dick, have a nice evening. So we're going to sign off now. Thank you.